Good morning to everyone. Myself Roja, Assistant Professor in Department of Microbiology, Dhanlakshmi Srinivasan Arts and Science College for Women Autonomous. Now we will discuss about antigen and antibodies. Introduction about antigen: substance capable of inducing a specific immune response is known as antigen. Immunological properties of antigens. Antigens can be defined on the basis of 4 immunological properties. First one is immunogenicity, second one is antigenicity, third one is allerogenicity, fourth one is tolerogenicity. What is immunogenicity? Ability to induce humoral and or cell-mediated immune response is known as immunogenicity. In this immunogenicity, B cells combine with antigen, then enhance the plasma cells and memory cells. The T cells combine with antigen and MHC, major histocompatibility complex, then give T effector cells and memory cells. In this context, we can refer to an antigen as an immunogen. Second one is antigenicity. What is antigenicity? Ability to combine specifically with the final product of above responses, that is antibodies and our cell surface receptors. All molecules immunogenic are antigenic. Reverse is not true. Example, haptens, molecules that can bind to antibodies or surface receptors. However, they cannot induce specific immune response alone, so known as non-immunogenic. Third one is allerogenicity. What is allerogenicity? Ability to induce various type of allergic reactions. What is allergens? Allergens are immunogens that specifically activates humoral and or cell-mediated responses with allergic manifestations. Fourth one is tolerogenicity. Capacity to induce specific immunological non-responsiveness in either humoral or cell-mediated responses. Tolerance induction is learned or acquired. It is induced more rapidly in immature lymphocytes than in mature ones. Maintenance of tolerance depends on persistence of antigen. Both B and T cells display tolerance. They differ in quantity of antigen needed to induce tolerance and duration of tolerance state. Experimental models, tolerance more effectively maintained in the T cells compared to B cells. What are the factors influencing immunogenicity? Immune system protect against bacterial, viral, fungal and parasitic infections. It recognizes macromolecules in the structure of these infectious agents, that is proteins or polysaccharides. Proteins are most potent immunogens with polysaccharides ranking second. Lipid and nucleic acid do not serve as immunogens. In cell-mediated immunity, only proteins serve as immunogens, must be processed to small peptides in association with MHC molecules. Immunogenicity is not intrinsic property. Immunogen contribution to immunogenicity. Immunogenicity is determined by 5 properties of immunogen. First one is foreignness, second one is molecular weight, third one is chemical composition, fourth one is complexity, fifth one is degradability. Foreignness, ability of lymphocyte to recognize self-antigen occurs during maturation. Any molecule not exposed to immature lymphocytes during this critical period is non-self or foreign. Degree of immunogenicity depend on degree of foreignness. The greater the phylogenetic distance between two species, the greater the genetic and antigenic disparity between them. Bovine serum albumin, more immunogenic in chicken than goat. Exceptions, some macromolecules that are conserved cross species. Sperm or corneal tissue are very sequestered for immune system. In sperm or corneal tissue were to be injected into the same animal from which they originated, that is immunogenic. Molecular weight of 1 lakh Dalton, the best molecular weight, greater than 500 to 10,000 Dalton are poor. Chemical composition and heterogeneity. Synthetic homopolymers, single amino acids or sugar, lack immunogenicity regardless of molecular weight. Copolymers composed of different amino acids are immunogenic. Addition of aromatic amino acids, example tyrosine or phenylalanine, increases immunogenicity. Primary, secondary, tertiary and quaternary structure of proteins affect immunogenicity. Fourth one is degradability. Macromolecules that cannot be degraded and presented by APC are poor immunogens. Polymers of D amino acids cannot be degraded by macrophage enzymes, therefore poor immunogens. Large insoluble macromolecules more immunogenic than small soluble ones. Why more readily phagocytosed and processed? Contribution of biological systems to immunogenicity. A genotype of the recipient animal. mouse 1 and that antigen X level of antibody, the mouse 1 antigen X level of antibody, cross F1 generation, intermediate level of antibody, genotype of the recipient animal continued, immune responsiveness was then mapped to a sub-region of MHC. Proteins encoded by MHC play a central role in determining degree of immune responsiveness to antigen. Immune response influenced by genes encoding. First one is B and T cell receptors. Second one is proteins involved in immune regulatory mechanisms. Immunogen dosage and route of administration. Low dose failure to activate enough lymphocytes or induces non-responsiveness. High dose lymphocytes enter non-responsiveness state. Single dose usually not enough to induce reaction. Require repeated doses over a period of weeks to induce a strong immune response. Repeated dosing, that is known as booster dose, increase clonal proliferation of antigen-specific B cells or T cells. Common administration routes are intravenous, intradermal, subcutaneous, intramuscular, and intraperinatal. Route of administration determines which immune organ and cell population will be deployed. Intravenous administration induce carried first to spleen. Subcutaneous administration move first to lymph nodes. This generated difference due to differences in residing population of cells. Thank you. Thank you to everyone.